All right, thanks for having me. Yeah, I've been working here at the HIV databases since 1995 with Betty Korber. Betty's been the PI for the immunology side of the database and also sequence database, and I've been PI recently for the sequence database.
But if you mutually agree,
Um,
it's hard, isn't it? It's when they don't mutually agree that the issues arise.
and
And
our
I think
database integrates both, the sequences with immunology. So we track, how the virus is evolving within a given patient based on that patient's immune system. So it's really a unique system. Most other viruses don't have a lot of data in that line. So there's a picture of me because I can't have a camera here at the lab. And that's our home page. So you see sequence database, immunology database, and the other viruses goes to human hemorrhagic fever viruses and a few other viruses, hepatitis C virus, for example. But most of those we don't maintain as much as HIV. So today I'll be covering inter-subtype recombination and circulating recombinant forms, and not so much talking about unique recombinants or intra-patient or intra-subtype recombination, but I'll mention that a little bit. I'll talk a little bit, just a brief introduction of why retroviruses recombine, and then I'll get into the tools, and I'll mostly be covering one particular CRF, circulating recombinant form, because it's a good example. If you want to interrupt me with some questions as I go, that's fine. So HIV and all retroviruses, they package two copies of the RNA genome. It's a linear strand, and they put two copies into each virion. So this, in most cases, each single cell is infected with a single virus. So you have these what we call homodimeric genomes. The two copies of the genome are essentially identical to each other. But occasionally you'll get two viruses with slightly different or even very different genomes going into a single cell. And that gives you the opportunity then to package two different strands when the virus is budding out. Recombination, in this case, happens when the polymerase, the reverse transcriptase, switches strands between these two RNA templates that are in the cell. So there isn't, in most recombination in mammalian genomes, and so on mammalian chromosomes, or even bacterial chromosomes, you have strand breakage and rejoining in order to get different DNA molecules to recombine. In this case, it's a template switching during a reverse transcription that creates the recombinant genome. And the theory is that the reverse transcriptase will only switch strands maybe once or twice in this replication cycle. So it's not going to scramble up the two genomes. But within a patient, recombination can happen every day. So there's the potential for scrambling genomes over time within a single patient. So if a single patient is infected with subtype B and subtype C, in theory, they could create virions after years of infection that were scrambled between B and C. But so far, we've never observed that. We've never seen a scrambled up genome. Most of our recombinant genomes we see with inter-subtype recombination, they have a few breakpoints, maybe 6 or 7 or 8 segments, 9 or 10 segments of the two different subtypes scrambled. And quite often we see genomes that are kind of half subtype B and half subtype C, or half subtype A and half subtype D. So the scrambling doesn't seem to happen, and we don't know why. But we would be able to recognize and know a scrambled genome if we did find one, and we haven't found one yet. So, we've been studying this virus for 30 years, haven't found one. It's probably not going to happen. It probably has to do with compatibility of the different regions of the genome. Subtype A, subtype D are compatible enough to recombine, but not every recombinant is equally fit and going to survive and spread to other humans. So anyway, if you have recombinants, you'd like to detect those. Because they interfere with phylogenetic analyses and many other types of analyses. In a phylogenetic tree, a recombinant comes out in between the two clades. So an AD recombinant will be somewhere in between A and D, and it doesn't, either go with D or with A. And it's not a new subtype or a new virus, it's just an AD recombinant. So RIP stands for Recombination Identification Program, and that's one that we developed here at Leno 20 years ago. And it has good points. It's got an adjustable window size, pre-built test set. It allows you to input your own test set if you want. It allows you to select which subtype reference sequences go into the analysis. You don't get a plot with 40 different subtypes in there, which gets confusing. You can choose just the 3 that you think are pertinent to your sample. The cons on that one are it doesn't produce output with numerical breakpoint locations, and it's not very automatable. It's not a good one for submitting thousands of short reads into it. Jumping Hummer at GOBIX is nice because it uses a Hummer model, hidden Markov model of each subtype of HIV, A, B, C, D, and some CRFs. And it can give you a better statistical significance of exactly where it thinks the breakpoint is, and it does also number that breakpoint against the standard reference HXB2. The cons of that program are that it doesn't have all of the CRFs in there. For many of the CRFs, we only have 3 or 4 genomes, so we don't, there's not a very good model for a genome when you only have 2 or 3 sequences. A Hummer model is basically it's sort of like a consensus of each subtype, but it's more statistically robust than that. It tells you which sites are strongly indicative of subtype A or subtype B. The Rega genotyping tool is nice because it will accept hundreds of sequences. You can send an input file that has a few hundred genomes or gene regions into it, and it will email you back the results in about a day or so. The window size isn't adjustable, so sometimes you'll miss small regions. I believe their window size is set at 400 bases, so if you have a little 100 base chunk of subtype A placed into a subtype G genome, it may miss that. But it's got some nice output. It's got very extensive output, and it does boot scanning along the genome, which is nice. And then I'm not going to go into all the other tools. There are hundreds of them, actually, but I will be a little later in my talk spending a little bit of time with this NCBI genotyping tool, and that one I like a lot because it's so quick and easy to use. It's kind of I call it quick and dirty because it uses BLAST to do the alignment, but the cool thing there is you don't need to pre-align your sequence to any reference set. You can send in an accession number of a sequence, or you can send in a blank sequence, a raw sequence, and it does all the alignment for you on the fly, and it's very quick and fast. Here's a page that I won't go into, but it's got dozens and dozens of other recombination detection tools listed. And I won't be presenting all those today, but most of them, we're all looking for the same thing. We're looking for different regions of the genome that are derived from different subtypes of the virus. So they're going to have different distances from the different subtypes. So you can, whether you're dealing with a phylogenetic tree or just a percent identity table, either one can want. And some of those methods, like a percent identity table against a small subset of sequences, a few from each subtype, can be the quickest and easiest way to send through millions of sequences in an hour and get them all at least in the right ballpark, if not totally correct. So that's what I just analyzed 7,000 Pangea envelope sequences. And, the first thing I did was just get them all aligned, toss out a few sequences that obviously couldn't be aligned well, eyeballed the alignment, thought it was pretty good. Then I built a tree, just a quick neighbor joining tree. I could immediately spot that most of the sequences were either subtype A, subtype D, or subtype C. And then I could start to individually look at these sequences that were falling in between the subtypes on my tree and looking for recombination in them. So rather than sending every sequence through a recombination tool in the beginning, I did that also, but I found looking at the tree and comparing it to my results from a RIP analysis was the best way to go for me. And deciding which tool to use depends, entirely on what your process or what your problem is. If you have sequences from a single patient, you probably want to get the complete genome assembled and then study that with several tools or something. But if you have millions of sequences, short reads from thousands of from different patients, you can't afford to be spending much time on each one, and you want something that's very quick and easy. And I'm proposing here at the HIP database that we'll come up with a pipeline where we first do it like a blast search against a subtype reference set, probably also including all of the CRFs, and then depending on the results of that blast search, we will then feed sequences that are possibly recombinant into the recombination analysis tools. But anyway, it's there's no simple answer for what's the best way to do it for every process. So HIV has, I forget the count now, I think it's 13 subtypes and subtypes if you include A1 to A6, BCD. There is no E complete genome because we found E as part of a CRF01, and we've never found what we could definitively prove would be a non-recombinant E virus. For other viruses, we've got some that are similar to that situation where we have a partly, say, subtype A and partly a new subtype, and we're not giving them new letters like L, M, and N, and O, and P, and so on, because we've changed the rules on subtyping as we go along is what's happened. So A, B, C, D, and E were defined before we had any real definition of what a subtype should be. By today's standards, say B and D would be subtypes. They are closer to each other than full subtypes are. They're similar to, the distance between A1 and A2. And then we have the circulating recombinant forms. Those are viruses that are not only recombinant, but they're spreading around the world as regular viruses now. They're not that each of these viruses is from dual infected patients that are out there. They're the progeny or descendants of a single dual infected patient that produced a virus that spread. So the definition of creating a new named circulating recombinant form or a new subtype for that matter is 2 complete genomes and at least 1 more partial genome. We'd like to have evidence of 5 or 6 or 20 viruses of the same type before we give it a name, but sometimes people get away with just 2 genomes. So here's an example of 2 different CRFs. CRF01AE was the first defined, and the yellow regions are subtype E, mostly an envelope, and most of the rest of the genome is derived from subtype A. And CRF04 is, I'm presenting it here just because it's a very complex one, where we've got these white regions that are undefined. We don't know what subtype they are. We've got regions that are sort of K-like and H-like. And although that's, that could almost be completely a new subtype, CRF04. At first it was proposed to be subtype I, but we decided that different regions of the genome were close enough to subtype A or subtype G or subtype K that it should be a CRF. So at the HIP databases, we have a whole page about the CRFs, and it's got links to the maps. It's got these view breakpoints, clickable links, which take you to a table that tells you the precise location of the breakpoint, so you don't have to try and read it out of this PNG image. We should probably do a better job of coming up with a table that's more computer readable than this web page is. But if you have any particular CRF, you can go to these tables and at least get the numbers out quickly. And always feel free to write to us if you need, if you for some reason need a whole table of the numbers in a better format, an Excel spreadsheet or whatever, we can put that together and send it to you. And it always helps to get input from our users out there. If you see a need for this or that tool or table or feature, that's the way we improve is by feedback from our users. So this is the NCBI Quick and Easy tool I was talking about. Comes preloaded with all of these different viruses. HIV1, they provide several different ref sets from something they started with in 2005 that we sent them actually, up to this 2009 ref set, which is the most recent they have and needs to be updated now because we have some new CRFs that they don't have yet. But anyway, this window over here, you can edit that list in that window very easily. You can add new sequences you want. You can change the labeling on these. If you wonder if yours is closer to this A2 than to that A2, you can make it A2A and A2B or whatever you want for your label. The window size is adjustable, so that's really quick and easy. You can start with a window size of 300, look at the result. You see something looks a little fishy, come back and maybe knock the window size down to 99 and have another look. I usually do that in multiples of 3. So if you have a window size of 300, use an increment of 100, window size of 150, increment of 50. Anyway, it's quick and easy to use, and you get great results. So here's an example of a complete genome. This would be a CRF03AB genome fed in, and I added a couple CRF03 reference sequences. And you can see across the complete genome, my query CRF03 tends to be most closely related to the existing CRF03 reference sequences. Over here, you see some funky dip in one of the references. It's probably just a slight misalignment. That's the weak point of BLAST alignments, is they don't do good with gappy regions. They don't do good with insertions, deletions. BLAST is prone to misaligning things a little bit. Another interesting feature of it, you see that it's the yellow here is the A6 virus, which is a subtype of A that's circulating in the Soviet Union, and that's where this recombinant formed, was in the Soviet Union. So you can see that the A6 viruses are as close to the average CRF03 as they are to each other. And then you see these regions where the light blue comes up, and that's subtype B. So we have an AB recombinant, but it's much closer to this A6 than it is to your average B from around the world. If we put in some Soviet Union B, we could probably get, some lines up here indicating that it was from a Soviet Union subtype B. And in this case, that's a window size of 150 and a step of 50, and you can see that there's, the lines are a little bit noisy. If you go to a window size of 300, you get a smoother curve sort of thing, but you lose these dips and peaks that may be important. Now this is a similarity plot, which is essentially the same thing as the window blast, but to do a SimPlot, you have to create your alignment yourself and feed that alignment into the SimPlot tool. And in this case, I have included a B from Georgia, former Soviet Union region. See that dark blue, and now it is up there, going right with the CRF03s, which are in green. And here I've got, several sequences of each subtype included. And so it's a little messy, but it's easy in Simplot to color the ones you're interested in, either by clicking, whoops, sorry, I didn't mean to do that. Either by clicking on the lines in your Simplot, you can recolor them, or you can go over to this table and right-click, and so you can either recolor by name over here and see where in the plot they are, or you can recolor by plot and see which names those pertain to. So that can be really helpful when you're uncertain about what the heck you've got. Another problem with this position on a Simplot is normally you would gap strip your alignment before you put it into the Simplot, and then it's hard to do the HXB2 numbering because you've stripped out gaps and stripped out pieces of the HXB2 genome and screwed up your numbering. But you can get a rough ballpark figure of where the recombination sites are. And you'll see right here in the center of the HIV genome, there's this tiny little region that happens to be very low in phylogenetic signal. There's a particular reason why there's an identical conserved, like a 20-mer in there that's almost invariant in the genome. And it's also a site that's involved in replication of HIV, so it may tend to be a hotspot for recombination. But anyway, that one little region of the genome is something that's often interesting or funky, and you see it in this plot. So these are two different maps of CRF03AB. The publication describing it didn't decide to put in that little region in PolGene, PolVIF region, where it was sort of A-like. The jumping Hummer analysis also didn't rate that as deserving to be called subtype A. But I'll show you in a second that there really is a tiny region of subtype A stuck in there. So here's the publication on CRF03, and they did note that region in their similarity plot or boot scanning, both. This is a similarity plot at the top and a boot scan at the bottom. Excuse me, I'm gonna drink some water. So they noted in the publication, but they didn't put it in their map, and it didn't end up on the CRF page, but it's there. Here, I'm not zooming in on that funky region. I'm zooming in on the, I'll go back. I'm zooming in on this region right here where it goes from subtype A to subtype B in the POL gene, which is fairly conserved and a fairly clear breakpoint. And you can see up in this region, I've got all the A's above my recombinant and a bunch of B's down below my recombinant. And you can see over here, we've got some sites where the CRF03 is clearly A-like and not B-like. And over here, you can see some sites where it's clearly B-like and not A-like. And then in this region in the middle, we've got an uncertain region, largely just because that region is so conserved. But this is common to all recombinants, really, is that they tend to recombine in these conserved regions. I think there's probably some threshold of similarity or identity needed in order for the polymerase to decide it can switch strands. It's not going to, doesn't want to switch strands that aren't homologous, so it won't switch strands in very diverse regions. So you can, these are multiple sequence alignment viewers. AliView is free for Macintosh, and BioEdit is the equivalent for Windows. But it's really handy or critical to be able to view your sequences, sort them into, so you can put your recombinant in between the two subtypes you're interested in. It's critical to have an alignment viewer and to be good at using it. Even if you don't adjust the alignment by, edit the alignment with the viewer, you want to be able to color it differently, move your sequences around, do some statistics. As much as possible with an alignment viewer, you're actually looking at your data, and that's critical. With AliView, you can change, or BioEdit, you can change the coloring like this, and that tends to make these regions of identity or difference stand out a little bit more. And you can even select one of your sequences and show differences of that sequence, and that really suddenly makes things stand out. So here we can see that our query sequence is much more similar to subtype A here, and a lot of differences from B. Over in this region, it's really similar to B and a lot of differences from subtype A. So anyway, just by switching your view of your sequence data, you can spot problems, you can spot errors, you can spot recombination sites and so on by eyeball. And there's no substitute for looking at your data by eyeball. The other thing that the alignment viewer is good for is in the end when you want to say exactly where is that recombination site, you can have the HXB2 standard reference sequence included in your alignment. You can pick, whoops, sorry, I don't know why this goes to the next slide when I click on it. You can pick the site you want to say is the center of your recombination, and it tells you the position you're at in the sequence you're looking at. So here you can see I've selected the HXB2 standard, and it's telling me I'm at position 2704, and then I can put that into my table, and it ends up in my map. And conversely, if you have like the Jumping Hummer numbering to tell you where it thinks the recombination is, you can find that site by coming up with this number, finding it in HXB2, and seeing if you agree with what Jumping Hummer told you. So this is a Jumping Hummer result of a CRF03 AB recombinant. And again, you see it kind of came up as equivocal in that tiny little region, middle, fairly clear breakpoint at the other two sites. But then it tells numbering both against your query sequence that went in, which can be really handy for looking again at your sequence and figuring out where the breakpoint is, or numbering based on HXB2, which is good for publications and describing to other people where the recombination sites are. Then we provide this map drawing tool, and you can see down here I've put in that the central region is, AB unequivocal, so it comes up as a blue-red combo there. This is another similarity plot of CRF03, and here instead of using dozens of sequences of each subtype, I've got a consensus of A, a consensus of B, a consensus of C and D, and so on. And that can be a much more clear figure for a publication than putting in dozens of sequences. In this similarity plot, I just wanted to point out one thing, was there's a couple of funky little dips in it, and especially this one where you've got a CRF03, no, I guess that's actually the B Georgia genome, but it's coming along, it's looking like there's a sudden dip in it. Is that would be indicative of recombination possibly. So if you then go and look at your alignment in that region of the genome, it turns out that no, that was just misaligned. And it's only this tiny little chunk of misalignment here. I can shove it back to the left, and now you can see these T's and CT's match up better. And that takes that dip completely out of the plot. So the point here I wanted to make was that sometimes similarity plots can help you to find regions of misalignment or regions where the data may be a little questionable. You need to go back and forth between different views of your data to find little things like this. I would never spot this by eyeball scanning along the alignment, but seeing that in my plot all of a sudden said, oh man, I got to go look at that, and I could see that my alignment was slightly off. And little things like that can also have a huge impact on a phylogenetic tree, other analysis, because you're counting this as being like 10 mutations right there, when really there was a single event of a deletion of 3 bases. This is just zooming in on this recombination region that was sort of equivocal. You can see right here is where there's a low phylogenetic signal. Everybody tends to come up to be similar to each other in this region of the genome. But if you look at the alignment highlighting the CRF03, you can see it's much closer to the A's, and specifically the A6 viruses in that region. And this is BCD, HIJK, all of the other viruses, and it's not, it's sort of equidistant to all those others. So the most likely explanation, especially seeing is that we already know that this virus has some A6 regions in it, clearly, that this region is also derived from A6 and not just aberrantly similar to the A6. Building a phylogenetic tree of just this region to see what that looks like. And what we see is that the CRF03 is indeed in the A6 clade there, not just a little similar to it, but it's right there in the clade. Another thing you can see is that subtype B and subtype D are separated from each other in this tree. One of these D's is kind of going over with H. The F1 and the F2 are spread out in different places. There's F1 up here, and F2 is way down here. So what that tells you is that, that region of the tree has some phylogenetic signal. The F2s are going together, the A2s are going together, the Bs and the Ds are going together, but it's not a clear, good phylogenetic signal. You got F1 and F2 separated from each other, B and D separated from each other. You've got some long branches and short branches. When you see a tree that looks like this, it just tells you that the amount of data you have in there, the phylogenetic signal you have in that region is just a little bit weak. A lot of times I'll see publications where they say that, oh, look, these sequences are on a long branch, they evolve faster. And that's really just not right. It's mainly that there just wasn't enough data there. You've got stochastic noise, a little bit of change, a couple of single base changes make a huge difference in your branch lengths. This is just showing the Rega subtyping tool where you can either paste in an individual sequence or you can upload a file of sequences. Now, there are other tools like Highlighter here that they're a little bit, this one's a little bit similar to when I was showing you the alignment and coloring sites that are changed or different. But this is more sophisticated. You can get silent versus non-silent sites highlighted. You can get hypermutation sites where a G to A transition is being influenced by ApoBec. And so on. You can get more things highlighted. And it's also clean and simple and a good publication figure. But anyway, this patient was at least dual infected. And you can see that when you have a virus that has this pattern with a little bit of change in the middle and then only very slight changes on the end, that's from recombination of a chunk of this virus being recombined into something of your consensus or top virus that you put in. So anyway, this is good for very small distances within a subtype and especially within a patient. It's good for highlighting sites that are being driven by an immune system. You'll see something like this where all the viruses are getting mutated in one region due to a MHC allele attacking it there. And it's good for looking for intra-subtype recombination. And then we have this new program just out a few weeks ago now called RAPPER, Recombination Analysis Program. And this again is for within a single patient. We're studying this patient over time, and you see that the diversity increases and you get all these viruses that look like they've evolved on their own. But when you go and look at the sequences, you find that... Do I have a question? No? Anyway, you find out that a lot of these clades can be similar recombinants or anyway, that they're derived from viruses that existed earlier. They're not just uniquely mutating to a new state. They're combining other genomes to make new forms. So like all of these viruses right here are a little bit of the red and a little bit of the blue. And these are not subtype ABC. This is time point ABC with this one patient. So that's the quick story. Does anybody have questions for me?